Welcome to this deep dive into the Rad Onc Smart Review RadBio edition.
Hey there.
Today, we're going to really get into something fundamental for anyone in radiation oncology.
Yeah, absolutely. We're talking about how radiation actually makes cells die. It sounds simple, maybe, but the mechanisms are, well, quite complex.
Exactly. We'll look at the different ways it happens, what it looks like, microscopically, and what's going on at the molecular level.
And understanding this stuff is just so crucial for knowing why treatments work, or sometimes why they don't work as well as we'd hope.
Right. So last time, we focused on how radiation initially damages DNA. Remember those double-strand breaks?
Mm-hmm. And the nasty chromosomal problems that come from them, like dicentrics and those ring chromosomes.
Yeah, those lethal aberrations.
And it's really key to connect that initial damage.
Mm.
That's the trigger, That's what kicks off all these different ways a cell can die or stop functioning, which is what we're focusing on today.
Perfect setup. So our plan today is to map out these main pathways. We've got apoptosis, that's the programmed cell death.
Right?
Then necrosis, and also its cousin, necroptosis.
Mm.
Mitotic catastrophe, death during division. Autophagy, which is kind of self-eating.
Yeah, a bit paradoxical sometimes.
And finally, senescence, where cells just stop dividing permanently.
And in telling these apart, understanding the differences, that's really key for interpreting treatment response.
Definitely. So let's kick off with apoptosis,
Yeah.
often called programmed cell suicide.
Right. That's the one. Apoptosis is, well, it's a remarkably orderly process. Think of it like a controlled demolition rather than just, blowing things up randomly.
Okay.
The cell basically activates a program to dismantle itself neatly. It's essential for normal development, like shaping our fingers in the womb, but also for getting rid of damaged or infected cells. And radiation is a big trigger for it.
And visually, under a microscope, it looks quite distinct.
Oh, absolutely. You typically see individual cells affected, not whole groups. The cell itself shrinks down.
Shrinks, okay.
The chromatin, which is the DNA packed with proteins it condenses, gets really dense.
Uh-huh.
Then the nucleus itself starts to break into fragments. And the cell membrane does this interesting thing called blebbing.
Blebbing, like little bubbles.
Exactly, little outward bulges or bubbles. And these blebs then kind of pinch off, forming what we call apoptotic bodies. Apoptotic bodies.
Got it. Apoptotic bodies.
And these are like neat little garbage bags containing the cell's insides. They get quickly eaten up, phagocytosed is the technical term, by neighboring cells or immune cells like macrophages. Phagocytost.
Okay, so it's a very contained process.
Yeah.
Now, what about the biochemistry? What's happening inside?
Well, it's an active process. It needs energy, ATP, and it involves turning on specific genes. The real stars of the show are a family of enzymes called caspases. K-A-S-pases.
Caspases, right. The executioners.
Pretty much. You have initiator caspases, like caspase 8 and 9, that get the signal and start the process. Then they activate the executioner caspases 3, 6, and 7 are the main ones, and these guys go around chopping up key proteins in the cell, leading to that dismantling we talked about.
And they chop the DNA too, right. But in a specific way.
Exactly. It's not random shredding like in some other forms of death. Enzymes called endonucleases cut the DNA specifically between the nucleosomes. Those are the spool-like structures DNA wraps around.
Okay.
So if you take that DNA and run it on a gel, you don't get a smear. You get this very characteristic ladder pattern. Fragments in multiples of about 185 base pairs. It's a classic hallmark.
Wow, okay. A DNA ladder. Anything else biochemically important?
Yes. Another key signal is that a lipid molecule called phosphatidylserine, FOSS, photidylserine, which is normally on the inner side of the cell membrane, gets flipped to the outside.
Flipped to the outside. Why?
It acts like an eat-me signal. It tells those phagocytic cells, hey, this cell's undergoing apoptosis, come clean it up. And we can actually detect this using a protein called annexin 5 in the lab.
Fascinating. And because everything is neatly packaged and cleaned up?
Right. There's usually very little inflammation associated with apoptosis. It's a clean death, doesn't really alarm the neighbors, so to speak.
Makes sense. So what actually flips the switch? What triggers apoptosis?
Two main routes, generally. There's the intrinsic pathway, which is often triggered by internal stress, like, importantly for us, DNA damage. This can activate the famous p53 protein, the guardian of the genome, which then turns on pro-apoptotic genes. Things like PMO, PYOMA, and XNOCASU and BX are key players here. They work at the mitochondria to release other factors that activate caspases.
Okay, so internal damage signals, what's the other way?
The extrinsic pathway.
No.
This involves signals coming from outside the cell. Specific molecules, ligands like Fas ligand or tumor necrosis factor alpha, bind to death receptors on the cell surface.
Death receptors, okay.
And this binding directly activates initiator caspase 8, kicking off the cascade from the outside in.
Gotcha. So tying this back to radiation.
Ionizing radiation is a really potent inducer of apoptosis, mostly working through that intrinsic pathway because of the DNA damage it causes.
Right.
But, and this is super important clinically, how readily cells undergo apoptosis after radiation varies hugely between cell types.
Ah, okay. Like what?
Well, hematopoietic cells, hematopoietic, those are blood-forming cells, and lymphoid cells, like lymphocytes, they're very sensitive. They'll often undergo apoptosis quickly, even at lower radiation doses. Think lymphomas and leukemias.
Okay. Highly sensitive.
But many solid tumors, especially things like sarcomas, they tend to be much less reliant on apoptosis. They might die, but often through other mechanisms we'll get to, so you can't assume radiation always kills just via apoptosis.
That's a critical distinction. Okay, so if not always apoptosis, what's next?
Yeah.
Necrosis.
Yeah, let's talk necrosis necrosis. This is kind of the polar opposite of apoptosis in many ways. It's generally thought of as accidental or uncontrolled cell death.
Accidental, like from severe injury.
Exactly. Things like severe lack of oxygen, toxins, physical trauma, or maybe very, very high radiation doses might cause it. It's more like catastrophic failure than a planned shutdown.
And it looks different too, I assume.
Definitely. Instead of single cells shrinking, necrosis often affects patches of cells. And instead of shrinking, the cells swell up. That's called oncosis, ONGKOHSIS.
Swelling, okay.
Their insides, the organelles, also swell and start breaking down. Eventually, the cell membrane just gives way, it ruptures, and everything spills out. That's lysis, LYSS.
Spills out. That sounds messy.
It is. Biochemically, it's mostly passive. The cell's energy systems fail, ion pumps stop working, water rushes in, hence the swelling. No complex signaling pathways needed usually.
And the DNA. No ladder.
Nope. The DNA breaks down more randomly, so if you run it on a gel, you just see a diffuse smear, no neat ladder pattern.
And because everything spills out. Inflammation.
Huge inflammation. All those intracellular contents spilling into the tissue act as danger signals, recruiting immune cells and causing a significant inflammatory response. Big difference from apoptosis.
Right. Now, you mentioned necroptosis earlier. Necroptosis. How does that fit in?
Yeah, necroptosis is interesting. It's a more recently understood pathway that's basically programmed necrosis.
Programmed necrosis. So it looks like necrosis, but it's regulated.
Exactly. Morphologically, it's necrosis, swelling, rupture, inflammation. But it's triggered by specific signals, often involving those same death receptors like TNFR1. But crucially, it happens when the apoptosis pathway, specifically caspase activity, is blocked.
Ah, so if apoptosis is prevented, this pathway can kick in.
Kind of, yeah. It's caspase independent. Instead, it relies on these kinases called RIPK1 and RIPK3 receptor interacting protein kinase 1 and 3. They trigger the necrotic-like death.
Okay. And is it relevant to radiation?
It's thought to play roles in things like immune defense against viruses, maybe some inflammatory diseases. Its exact role after radiation is still being actively researched, but it's another potential way cells might die, especially if apoptosis is inhibited.
Interesting. Okay, so we have the clean death, apoptosis, and the messy death, necroptosis. What about mitotic catastrophe? Catastrophe.
Right. Now, this one is hugely important for radiation oncology, especially for solid tumors treated with standard doses.
Why is that?
Because mitotic catastrophe isn't really a distinct biochemical pathway like apoptosis. It's more of a consequence, a mode of death that happens when a cell tries and fails to divide properly because its chromosomes are too damaged.
Ah, so it links directly back to that initial radiation-induced chromosome damage we discussed, the decentrics, rings, breaks.
Precisely. A cell gets irradiated, its DNA gets damaged, maybe some lethal chromosome aberrations form. Then, if the cell cycle checkpoints fail, which they often do in cancer cells, the cell tries to go through mitosis, the M phase.
Tries to divide with messed up chromosomes.
Yep. And it's a disaster, hence catastrophe. Under the microscope, you see cells struggling through division. The chromosomes don't segregate properly.
What does that look like?
You often end up with cells that are abnormally large, or cells that have multinucleation. Or you might see little extra blobs of nucleus called micronuclei, which contain chromosome fragments that got lost during the messed up division.
Wow.
Ultimately, the cell fails to complete mitosis successfully. It might die during mitosis or shortly after in the next phase. And this failure to divide successfully is essentially what we measure as reproductive cell death in our standard lab assays, those clonogenic assays.
So mitotic catastrophe is a major reason why radiation stops tumor cells from forming colonies.
For many solid tumors at clinical doses, yes, it's considered a primary driver of reproductive death. It's like, trying to build two identical houses using a blueprint that's been completely scrambled and torn up. It just doesn't work.
Great analogy. Okay, that's mitotic catastrophe. Now, what about autophagy? Autophagy. You said it was paradoxical.
Yeah, autophagy is fascinating. Think of it as the cell's internal recycling system or quality control process.
Recycling.
The cell essentially eats parts of itself. It engulfs old, damaged organelles or misfolded proteins within these double membrane sacs called autophagosomes. Autophagosomes.
Okay.
These autophagosomes then fuse with lysosomes, the cell's waste disposal units, forming autolysosomes, AW to L lysosomes. Inside there, enzymes break down the contents, and the basic building blocks can be recycled and reused by the cell.
So that sounds helpful, like cleaning house.
It often is. That's the pro-survival side. If a cell is stressed, say starved of nutrients, autophagy can break down non-essential bits to provide energy and materials, helping the cell survive tough times.
Okay, but you said it can be pro-death, too.
Yes. Sometimes, if autophagy is excessive or prolonged, this self-eating can go too far and actually lead to the cell's death. We call that autophagic cell death. It's like the recycling program gets out of control and starts consuming vital machinery.
A double-edged sword, then. What controls it?
It's complex, regulated by many proteins. A key one is Beclin-1, BE-Kalin-1, and there's a whole family of ATG genes, autophagy-related genes. It's often inhibited by a major growth signaling pathway called MTO.
And radiation.
Radiation definitely can trigger autophagy, but whether this ultimately helps the tumor cell survive the radiation stress, making it more resistant, or whether it actually contributes to killing the cell, that's a really hot area of debate and research. It seems to depend a lot on the cancer type, the context, the dose. It's complicated.
Right. Not a simple answer there. Okay, last one on our list. Cellular senescence. CNES.
Yeah.
You said this isn't technically cell death.
Correct. Senescence is a state of stable, long-term, essentially irreversible growth arrest. The cell stops dividing permanently, but it doesn't die. It's still metabolically active, just retired from the cell site.
Retired? Okay. What makes a cell decide to retire?
Various stresses can trigger it. Again, DNA damage from radiation is a big one. Another trigger is telomeres shortening. Those protective caps on chromosome ends get shorter with each division, and when they get too short, it signals the cell to stop dividing. That's called replicative senescence. Activation of certain oncogenes can also trigger it, surprisingly.
And pathways involved, similar to apoptosis triggers.
That often involves those same key tumor suppressor pathways, yeah.
P53, working with its partner P21, is a major route, and another is the P16 pathway acting on the retinoblastoma protein, RB. These pathways put the brakes on the cell cycle.
Do senescent cells look different?
They often do. They tend to become larger, flatter, kind of spread out, and their chromatin reorganizes into these distinct clumps called senescence-associated heterochromatic foci, or SAHFS.
Okay. And is there a specific marker we can look for? Yes. The classic biochemical marker is increased activity of an enzyme called senescence-associated beta-galactosidase, GU-LK-SY-dase. We usually call it SA-beta-gal. You can stain for it. And it shows up specifically in senescent cells when you do the test at a slightly acidic pH 6.0.
SA-beta-gal at pH 6. Okay, but if they're not dead and not dividing, what are they doing? Just hanging out?
Not quite. That's another really interesting part. Senescent cells often start secreting a whole cocktail of molecules, inflammatory cytokines, growth factors, enzymes that remodel the tissue around them. This mix is called the senescence-associated secretory phenotype, or SASP.
SASP. And that sounds like it could have big effects.
It really can. The SAP can be a mixed bag. It can alert the immune system to clear damaged cells, which is good.
Yeah.
But it can also promote chronic inflammation, and in some contexts, maybe even fuel nearby tumor growth or fibrosis. It's complex.
So from a radiation perspective, inducing senescence in a tumor cell, that counts as controlling it, right? Because it can't divide anymore.
Exactly. Loss of reproductive capacity is our goal, so inducing senescence contributes to tumor control, just like causing outright death. It's part of that cell kill we measure. But senescence induced in normal tissues might also contribute to some late side effects of radiation therapy.
Right, that makes sense.
Yeah.
Okay, before we move to clinical pearls, just quickly imaging. Can things like PT, MRI, CT scans tell us how the cells are dying?
Not directly, no. They show us changes in the tumor's size, metabolism, structure. But we might get indirect clues, like if a tumor shrinks really fast on a CT or MRI after radiation.
Maybe more apoptosis. Because it's quicker and cleaner removal.
Potentially, yeah.
Yeah.
Whereas a slower response, or maybe changes suggesting inflammation, might hint at other mechanisms like necrosis or maybe cells undergoing senescence or mitotic catastrophe before eventually dying or being cleared. It's inferential, though. Same goes for pathology, looking at tissue samples. You don't directly assay for the pathway usually, but the patterns of cell loss or morphology might give clues in the context of AJCC staging and response assessment.
Got it. Indirect clues, not direct visualization of the mechanism. Okay, perfect time for some clinical pearls. What are the key takeaways for practice?
All right, pearl number 1. Apoptosis is programmed generally non-inflammatory. That makes it, in theory, a really desirable way to kill cancer cells, right? Minimal collateral damage to healthy tissues.
Yeah, the ideal scenario. Pearl 2.
Pearl number 2. Remember mitotic catastrophe. That death linked to failed cell division from chromosome damage. That's a major mechanism for how radiation kills cells in many solid tumors at the doses we typically use, measured in gray, GI, or centiGray, CA-GI. Don't underestimate its importance.
Crucial for solid tumors. Okay, and pearl 3.
Pearl number 3. Different tumors have different tendencies. Lymphomas might heavily rely on apoptosis. Sarcomas, maybe much less so, relying more on mitotic catastrophe or senescence. This variability helps explain why different cancers respond differently to radiation and might need different dose schedules or combinations.
That really ties together clinically. And thinking about those differences, does that open doors for, say, clinical decision-making? Tailoring treatment.
Absolutely. That's where a lot of research is heading. If we know a particular tumor type is, say, resistant to apoptosis, maybe we can add drugs that target that pathway. For example, there's a lot of interest in drugs targeting anti-apoptotic proteins like BCL2. The idea is to lower the threshold for apoptosis, making radiation more effective.
Makes sense. Trying to push cells towards a more favorable death pathway.
Exactly. Or maybe finding ways to exploit senescence or autophagy, although those are trickier, as we discussed.
And do our fancy treatment techniques like IMRT, VMAT, SBRT, SRS, do they change which mechanism happens?
Not directly. The technique itself doesn't dictate the biology, but the dose and the fractionation, how we deliver that dose over time, certainly can. Very high doses per fraction, like an SBRT or SRS, might cause more direct damage, leading perhaps to more necrosis or faster apoptosis in some sensitive cells, alongside mitotic catastrophe. Standard fractionation might rely more heavily on accumulation of damage, leading to mitotic catastrophe or senescence over time. So dose and schedule influence the balance of these mechanisms, potentially.
Right, the dose matters.
Oh.
Okay, ready for a quick board blitz? Test our recall.
Let's do it. Fire away.
Question 1. Which form of cell death is known for cell shrinkage, membrane blebbing, caspase activation, and that specific DNA ladder pattern with fragments around 185 base pairs?
Okay, shrinkage, blebbing, caspase's DNA ladder. That screams apoptosis.
Correct. Apoptosis, all the classic signs.
Yep.
Question 2. A researcher looks at irradiated cells days later. They see lots of really big cells, some with multiple nuclei, others with little micronuclei. What's the most likely mode of cell death here?
Big cells, multiple nuclei, micronuclei. That sounds like division gone wrong. Mitotic catastrophe.
Exactly. Mitotic catastrophe. Hallmarks of failed mitosis after damage.
Right.
Question 3. Increased activity of a specific enzyme, SA-beta-gal, measured at pH 6.0, is a marker for what cellular state?
SA-beta-gal at pH 6, that's the specific marker for senescence, the cells that stop dividing.
Correct again, senescence.
Okay.
Last one, question 4. Which type of cell death typically involves cell swelling, membrane rupture, release of cell contents, and a strong inflammatory response?
Swelling, rupture, inflammation. That's the messy one. That's necrosis.
Perfect. Necrosis, the uncontrolled inflammatory death. Okay, great run through. Now, briefly, any major controversies or advances we should touch on?
Well, we hinted at it, but the role of autophagy is definitely still debated and actively studied. Is it pro-survival, helping tumors resist treatment, or pro-death? And can we target it effectively? There's a lot of work trying to figure out how to manipulate it, maybe inhibit it to sensitize tumors or push it towards cell death.
Yeah, that complexity is definitely a frontier. Anything else?
Senescence is another big one. We're learning more about the SASP, that secretory phenotype. While inducing senescence stops tumor growth, could the SASP sometimes cause problems later, like inflammation or even promoting other nearby cells to grow? So there's interest in senolytics drugs that might clear out senescent cells or ways to modify the SASP. It's a rapidly evolving area.
Very interesting. Managing the consequences of stopping division. Okay, so let's wrap up with a quick summary of the key features.
Sounds good. So, apoptosis, programmed, needs caspases, clean DNA cuts, ladder, membrane blebbing, non-inflammatory cleanup.
Necrosis, messy, accidental swelling, lysis, inflammation, random DNA smear.
Hmm.
And its regulated cousin, necroptosis, programmed necrosis, inflammatory but caspase independent, uses RAP kinases.
Right. Mitotic catastrophe. Death from trying to divide with damaged chromosomes, think multinucleation, micronuclei, a major killer in irradiated solid tumors leading to reproductive death.
Autophagy, cell self-eating recycling, can be pro-survival or pro-death, involves Beclin-1 and ATG proteins. Complex role.
And finally, senescence. Irreversible growth arrest, not death. Marked by SA beta-gal. Cells are alive but retired and can secrete that SASP cocktail, which has downstream effects.
Excellent summary. And the big picture takeaway.
Is that knowing these pathways isn't just academic, right? It's fundamental to understanding radiation effects, predicting response, figuring out resistance, and ultimately designing better cancer treatments in the future. It connects the bench to the bedside.
Couldn't agree more. It's the foundation.
Definitely.
Well, that brings us to the end of this deep dive. To really hone your skills and test yourself with practice oral boards, check out radonsmartlearn.com.
And make sure you subscribe to Rad Onc Smart Review so you don't miss our next exploration.
Thanks so much for tuning in.